Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Right now, across the globe, there's approximately 240 million people who wake up every single morning.
Every single day.
Right, every day. They take their prescribed medication and they go about their lives carrying this staggering underlying reality.
And that reality is that those daily pills, as vital as they are to keeping them alive, will almost certainly never actually cure them. I mean, they're essentially trapped in a medical holding pattern with chronic hepatitis B.
They are, but what if a 24-week injection could what if it could turn off the virus's camouflage entirely?
Right.
Because we're looking at a newly published set of phase 3 clinical trials today, specifically the BeWell-1 and BeWell-2 trials from the New England Journal of Medicine, and they demonstrate exactly that.
It's honestly a massive shift. A finite 24-week treatment successfully triggered what they call a functional cure in up to 28% of a very targeted group of patients.
Which is huge.
It is. These individuals dropped their lifelong medications completely and their bodies actually held the line without relapsing.
So, welcome to today's deep dive. We're going to unpack the primary data of these trials, which were led by Dr. Jinlin Hao and Dr. Sengyi Lim.
And we're also bringing in a really grounding concurrent editorial by hepatologist Dr. Anna S. Luck.
Right. Because the goal here isn't just to, look at the pop-line success rates and move on. We want to understand the biological mechanisms for you.
Exactly. Like, how does this new drug Bepirovirsen actually operate? Why does the baseline level of viral debris in your blood change literally everything about your outcome?
And why waking up an exhausted immune system is, it's a breakthrough, definitely, but it's also a highly delicate medical tightrope.
Let's start with the baseline, though. To really grasp why going from 0 to a 28% cure rate in this specific group is such a monumental shift, you have to look at the exhausting reality of standard care today.
It's a grind.
It really is. If you have chronic hepatitis B right now, the most common treatment is nucleoside or nucleotide analog therapy. You'll hear us call this NA therapy.
Drugs like entecavir or tenofovir.
Exactly. And look, to be fair, they do their primary job really well. They suppress the replication of the virus,
which drastically lowers your risk of liver cirrhosis and, liver cancer down the line.
They're highly effective at suppression, yes. But suppression is not eradication. That's the key.
Right. The virus remains physically present inside the liver cells. It's just sitting there.
And if you stop taking that NA therapy before you hit a very specific and incredibly rare milestone, the virus almost always comes roaring back.
That milestone being the complete loss of the hepatitis B surface antigen.
Right. And if you don't hit that and you stop the pill, it can sometimes trigger a severe, even life-threatening hepatitis flare.
Let's use an analogy here for you because the word suppression sounds a bit too final, I think.
It really does. Think of standard NA therapy like constantly bailing water out of a leaky boat.
I like that.
Right. As long as you have your bucket and you keep bailing water over the side every single day, the boat stays afloat. You aren't going to sink.
But the fundamental problem hasn't changed at all.
Exactly. There is still a hole in the bottom of your boat.
And if you stop bailing for even a little while, the water level just rises right back up.
The hole is the virus persisting in your liver. And the daily bailing is that NA therapy. You have to keep doing it forever because those pills, they do absolutely nothing to actually patch the hole.
And the data on achieving that actual patch under standard care is incredibly bleak.
It's depressing, honestly.
It is. After 8 to 10 years, think about that, a full decade of continuous daily NA therapy. Only about 3% of patients naturally achieve that complete loss of the surface antigen.
3%. After 10 years. That's just wild.
It's almost negligible. Now, there is an alternative out there. It's called pegylated interferon, which is a weekly subcutaneous injection for 48 weeks. And it does have a slightly higher success rate, maybe 3 to 4% at the end of treatment, and sometimes rising to 8 to 11% after a few years of follow-up.
But from what I understand, it's rarely prescribed today, right?
Very rarely. The side effect profile is just so severe. You're talking about intense flu-like symptoms, deep depression, extreme fatigue.
So most patients and their doctors look at that, 11% best case scenario and decide the misery of the treatment just isn't worth the slim chance of a cure.
Exactly. Which leaves millions of people tethered to a daily bucket, bailing water, knowing the chances of ever putting the bucket down are practically zero.
And that right there is the exact clinical wall that researchers were staring at when they designed the B-Well-1 and B-Well-2 trials.
They weren't looking for a better bucket.
Right. They're looking for something that fundamentally patches the hole, or, at least alerts the Coast Guard to come fix the boat for you.
So let's break down how they actually tested this. The B-Well trials were 2 replicate, double-blind, placebo-controlled phase 3 trials.
Meaning this wasn't some tiny speculative pilot study.
Not at all. They spanned 29 countries across Europe, the Asia-Pacific region, and the Americas. They enrolled a total of 1,838 adult patients.
That's a massive sample size.
It is. Now, all of them had non-cirrhotic chronic hepatitis B.
Meaning the virus hadn't severely scarred their livers yet?
Correct. No severe scarring. And critically, their viral loads were already fully suppressed because every single one of them was actively taking standard daily NA therapy when the trial started.
Okay, so they're already stable on their standard daily pills. How did the new drug actually fit into that routine?
Well, they randomized the patients 2-to-1. 2/3 received this new investigational drug Bepirovirsen as a 300 milligram subcutaneous injection once a week for 24 weeks.
And the other third?
The other third got a placebo injection. But here is the crucial variable. Everyone kept taking their standard daily NA pills during this entire 24-week injection window.
Oh, okay, so they didn't swap one for the other. They're adding a new mechanism directly on top of the old one.
Precisely. They add the injections. Then, after the 24 weeks of injections finished, the patients stayed on just their daily NA pills for another 24 weeks.
Just to monitor things.
Right, to stabilize. So they eventually hit week 48 of the trial. And at that exact point, eligible patients were instructed to do pretty much the scariest thing you can do when managing this disease.
Completely stop taking their NA therapy.
Yes, put the bucket down entirely.
And wait to see if the boat sinks. That must be terrifying for a patient who's been on these meds for years.
I can only imagine. But the entire primary outcome of the study hinged on what the patient's blood work looked like at week 72.
Which is a full 24 weeks after all medications, including the daily NA pills, had been completely discontinued.
Exactly. The researchers were looking for a functional cure.
Okay, hold on. Let's push back on this terminology right now.
Sure.
Because the word cure is a very heavy, very loaded word to throw around in virology. Why is the medical community calling this a functional cure instead of just an absolute permanent cure? Because, if I hear cure, I assume the virus is 100% gone from my body forever.
And that's a very fair assumption. It's an important distinction to make. And it really comes down to the insidious nature of the hepatitis B virus itself.
How so?
Well, the virus is a master of hiding. When hepatitis B infects a hepatocyte, which is a liver cell, it actually deposits its genetic material directly into the nucleus of that cell.
Okay.
And it deposits it in a highly stable form. It's called cccDNA. Or covalently closed circular DNA.
cccDNA. Got it.
It basically integrates its own blueprint right into your liver's hard drive.
Oh, wow. So even if the blood looks completely clean on a test, that blueprint is still just sitting there in the liver.
Exactly right. Even when we test your blood and we find absolutely 0 surface antigens and 0 circulating viral DNA, that cccDNA can still persist, hiding deep within the liver cells, totally dormant.
Right. So if you call it an absolute or sterilizing cure, you're promising the patient that every single microscopic blueprint has been totally eradicated.
And we simply can't guarantee that right now. If a patient achieves this functional cure, but then, say, 20 years later, they undergo severe immunosuppression.
Like if they need an organ transplant or they go through aggressive chemotherapy?
Yes. In those cases, the immune system drops its guard, and that hidden blueprint can theoretically reactivate.
That sounds a bit like false advertising to call it a cure then. I mean, why settle for functional?
Because for 99% of everyday life, it functions exactly like a permanent cure. The major liver disease associations globally all agree on this specific target.
What are the exact metrics for it?
If you hit the metrics of a functional cure, which means your hepatitis B surface antigen, or HBsAg, is undetectable at less than 0.05 international units per milliliter, and simultaneously, your viral DNA is unquantifiable at less than 20 units.
And you sustain that?
Sustained for at least 24 weeks after stopping all treatment. If you hit that, your life changes completely.
How much does the risk profile change?
Drastically. The risk of the virus spontaneously relapsing drops to near 0. Your risk of transmitting the virus to anyone else halts entirely.
And what about the cancer risk?
The looming threat of developing liver cancer decreases significantly, even further than it does with just standard viral suppression.
So you're entirely off the medication, and your own immune system is holding the virus in permanent check.
Exactly.
It's essentially an armed standoff.
Yeah.
But your body has won the territory, and it's set up a permanent perimeter. And you don't have to take daily pills to maintain that perimeter anymore.
That is exactly it. So let's look at the headline results from the trials. Did Bepirovirsen establish that perimeter?
Right, let's get into the numbers.
In B-Well 1, 20% of the patients who received the drug achieved that strict functional cure at week 72.
Okay.
In the replicate B-Well 2 trial, it was 19%.
And the placebo group?
In stark contrast, in the placebo groups across both trials, exactly 0% of patients achieved it.
Wow. So going from literally 0 to roughly 1 in 5 is a massive statistical jump.
It really is.
But if we stop the analysis right there, we are missing the absolute most fascinating part of this research. Because the true breakthrough isn't just that 20% average.
No, it's not.
It's actually hidden in the baseline characteristics of the patients themselves. The drug doesn't work equally well on everyone right now?
Not at all. Its efficacy is wildly dependent on how much viral debris is floating around in your bloodstream before you even take that very first injection.
This is the crucial nuance for anyone listening. The researchers actually stratified the patients into 2 distinct groups based on their baseline antigen levels.
Specifically, their starting HBsAg levels.
Right. So walk us through those 2 groups.
Well, the lower stratum consisted of patients who started the trial with 1,000 international units per milliliter or less.
Okay.
And the higher stratum included patients with levels between 1,000 and 3,000 units.
And when you split the data between those 2 groups, the numbers diverge so dramatically, it almost looks like you're looking at 2 entirely different drug trials.
It really does. It's striking. For the patients in the lower stratum, the ones starting with 1,000 units or less, the functional cure rate didn't hover at that 20% average. It jumped up to 25% in B-Well 1 and an impressive 28% in B-Well 2.
So we're talking more than 1 in 4 patients.
Exactly. But for the patients in the higher stratum, those starting with over 1,000 units of circulating antigen, the success rate completely plummeted.
How low did it go?
It fell to just 10% in B-Well 1 and a mere 5% in B-Well 2.
Okay, why? I mean, structurally, why does starting with a higher level of this specific surface antigen basically break the drug's effectiveness?
It's a great question.
If the drug works by attacking the virus, why can't it just attack a bit more of it?
To understand why, we have to look at what that surface antigen, the HBsAg, actually does to your immune system.
Okay, lay it out for us.
HBsAg isn't just some inert piece of viral debris floating around. The hepatitis B virus is incredibly devious. It's very smart. It produces massive, overwhelming quantities of these surface proteins, far, far more than it actually needs to assemble new infectious viral particles.
Wait, so it's overproducing them on purpose?
Yes. It pumps these excess proteins into the bloodstream specifically to act as decoys.
Oh, kind of like a fighter jet deploying flares to confuse a heat-seeking missile.
That is the perfect analogy, precisely. Your immune system, specifically your virus-fighting T cells, they are the heat-seeking missiles.
Right.
But when the bloodstream is absolutely flooded with tens of thousands of these viral decoy proteins, the T cells get constantly triggered. They are firing at everything, all the time.
They're overwhelmed by the flares.
Over years and decades of chronic infection, this constant overstimulation leads to a biological state that immunologists call T-cell exhaustion.
T-cell exhaustion. That sounds exactly like what it is.
It is. The immune cells literally burn out. They become dysfunctional and they just stop fighting. They're too overwhelmed by the decoys to actually clear the truly infected liver cells.
So if you are in the higher stratum, starting with over 1,000 units, your blood is so heavily saturated with those decoys that your T cells are profoundly exhausted.
Right. The data strongly suggests that Bepirovirsen works best when the overall decoy burden is already relatively low.
That makes sense.
If you start with under 1,000 units, the drug can knock out the remaining decoys fast enough that your exhausted T cells actually have time to wake up, realize the camouflage is gone, and attack the actual virus.
But if you start in the higher stratum, the sheer volume of antigen completely overwhelms the drug's mechanism.
Exactly. A 24-week treatment window simply isn't enough time to clear all those decoys and reverse years of deep, deep immune exhaustion.
That is fascinating. So the drug really needs the immune system to act as a willing partner. If the partner is too exhausted, the drug fails.
Yes.
But let's look at the practical outcome for the patients here, beyond just the strict mathematical definition of a functional cure. Because there is a very important metric in this study regarding the discontinuation of that standard NA therapy.
Yes, there is. And it's very promising. Across both trials, 24% of all patients taking bepirovirsen, regardless of their starting stratum, successfully met the clinical criteria to stop taking their daily NA therapy at week 48.
They got to put the bucket down.
They did. And in the placebo group, 0 patients met the criteria to stop.
And here's the detail that really stands out to me. Out of all those patients who stopped their daily pills, even the ones who didn't quite hit the absolute mathematical floor to be officially labeled a functional cure.
Right.
Not a single one of them experienced a clinical relapse by week 72.
None. 0.
I mean, some of them had minor, unquantifiable traces of the virus still kind of blipping on the radar, but none of them got clinically sick.
Exactly.
None of them needed to rush back onto the daily pills to stop a flare-up. Their bodies, having been given this 24-week boost from the drug, were holding the line on their own. They put the bucket down, and the boat wasn't sinking.
It is undeniably encouraging, especially for that low antigen group hitting the 28% functional cure mark.
Absolutely.
But we do need to pivot heavily here, because the biological process required to actually clear this virus is intense.
Very intense.
Achieving this cure isn't like, taking a course of antibiotics for an ear infection and just feeling a bit tired for a few days.
No, not at all.
The body's reaction to bepirovirsen is significant. And the safety profile makes it very clear that this is a heavy-duty therapeutic intervention.
The adverse event data definitely reflects a powerful drug that is essentially violently shaking the immune system awake.
Let's run through those numbers.
Overall, 91% of patients taking bepirovirsen reported some kind of adverse event.
Now, to be fair, the placebo group reported 73%, but those are mostly mild things.
Right. But when you look at grade 3 events, which the medical community classifies as severe, requiring actual medical intervention, they hit 16% of the treatment group, compared to just 3% in the placebo group.
And breaking down those adverse events shows us exactly what the drug is doing to the body. Injection site reactions were the most common.
That hit about 53% of patients on the drug, right?
Yeah. Yes. And those were mostly mild. Localized swelling, redness, primarily in the first 4 weeks. But the systemic reactions are what require highly vigilant monitoring.
What kind of systemic reactions?
Well, the trials noted significant changes in renal function. Specifically, they saw decreases in the estimated glomerular filtration rate, or eGFR.
Which, for those who don't know, is basically a measurement of how efficiently your kidneys are filtering waste out of your blood. So a drop in eGFR means the kidneys are under stress.
Exactly. There were also notable drops in platelet counts. 10% of patients on the drug saw their platelets fall below 100,000 per cubic millimeter.
Which raises bleeding risks.
Right. And in 2 specific cases, the platelets dropped dangerously low, below 50,000, which required permanent discontinuation of the drug to prevent severe bleeding risks.
Okay, so kidneys and platelets are affected. But the most paradoxical side effect, and biologically I think the most interesting one, involves the liver itself.
The ALT spikes.
Yes. The trials recorded that 24% of patients on bepirovirsen experienced a spike in their ALT levels.
Now, ALT stands for alanine aminotransferase.
It's an enzyme found inside your liver cells.
Right. And normally when you see a massive spike in ALT on a routine blood test, it sets off major alarm bells.
Because it usually indicates liver inflammation or injury or severe toxicity.
The cells are literally breaking open and spilling this enzyme directly into the blood.
And in the treatment group, these levels were sometimes spiking to at least 3 times the upper limit of normal.
If you just look at that raw data on a spreadsheet, it looks like the drug is destroying the liver.
It sounds terrifying.
Yet, the timing of these ALT spikes provides the most critical biological insight of the entire trial.
Because the spikes weren't happening immediately after taking the drug.
No, they weren't. The data shows these spikes occurred primarily between weeks 5 and 10 of the treatment window.
And that timing is key.
It is. When the researchers overlaid the graphs of these ALT spikes directly on top of the graphs mapping the patient's viral loads, they found a perfect inverse correlation.
Meaning what?
Meaning the liver enzymes were spiking at the exact same time the hepatitis B surface antigens were plummeting, often dropping by more than 3 logarithmic scales.
And this is where immunology is just wild. You have to look at this side effect not as toxic chemical damage from the drug itself.
Right, it's not the drug poisoning the liver.
It's battlefield collateral damage.
Exactly.
As the drug shreds the virus's camouflage, clearing out all those decoy flare proteins we talked about, the body's T cells finally wake up from their exhaustion.
They snap out of it.
They look around, realize the liver is absolutely full of infected cells, and they go to war.
They start systematically destroying the infected hepatocytes to clear out those hidden viral blueprints.
Exactly. The ALT spike is literally the biochemical smoke rising from the battlefield. The inflammation isn't a sign that the drug is toxic. It's a sign that the body is finally fighting back after years of just letting the virus hide in plain sight.
It is exactly what needs to happen to achieve a functional cure.
But that biological reality creates an incredibly tricky clinical tightrope.
It really does.
Because while the immune system needs to destroy those infected cells to clear the virus, you cannot let it destroy too many cells at once.
Or the patient will go into acute liver failure.
Exactly. The inflammation is necessary, but it has to be rigorously controlled.
Which means you can't just hand a patient a box of syringes and say, good luck, see you in 6 months.
Absolutely not. This therapy demands exceptionally rigorous patient monitoring.
The trial protocol actually required patients to physically go to a clinic and be monitored every 1 to 2 weeks.
They were getting constant blood draws.
Constant. If their liver enzymes, their kidney markers, or their platelet counts cross specific safety thresholds, the medical team had to immediately pause the injections.
The clinical management for this must be exhausting for the healthcare providers.
I'm sure it is. The reassuring part of the data, though, is that when the drug was either paused or finally completed, these alarming markers, the kidney filtration rates, the platelets, the liver enzymes, they generally returned to near baseline levels by week 72.
Okay, so the damage wasn't permanent.
No, not permanent, but managing that 24-week window requires a highly responsive medical team. They have to be ready to interpret real-time data and adjust the dosing schedule on the fly.
And that level of required monitoring leads us directly into the limitations of this study. Because honest science means acknowledging who wasn't even in the room for these trials.
Right, the exclusion.
The B-Well trial specifically excluded several major demographics. For instance, they excluded anyone with HIV or hepatitis C co-infections.
And as we discussed earlier, anyone with an antigen level over 3,000 was effectively non-responsive anyway, so it's not really a viable option for them right now.
But most importantly, they explicitly excluded patients who already had liver cirrhosis.
And that exclusion is deeply tied to everything we just discussed about the ALT spikes and the battlefield in the liver.
Make the connection for us.
If a patient already has a severely scarred cirrhotic liver, they have very little healthy tissue left to spare.
Right.
We simply do not know if their compromised liver could safely survive the intense immune system flare that this drug intentionally induces.
The collateral damage might push a cirrhotic liver completely over the edge into total failure.
Exactly. So we cannot generalize these 28% success rates to everyone living with chronic hepatitis B. It is a highly selected, relatively healthy population within the broader disease demographic.
To pull all of this into a broader global perspective, the concurrent editorial by Dr. Anna S. Locke is incredibly grounding.
She is a powerhouse in this field.
She really is. She's a leading hepatologist from the University of Michigan Health. She's co-authored the American Association for the Study of Liver Diseases guidelines on hepatitis B since, 2001?
Yes, and she helped write the very first World Health Organization guidelines back in 2015.
So she's looking at the entire global chessboard of this disease, not just one trial.
And Dr. Locke describes the B-Well trial results as a major step toward a functional cure, but she is very quick to acknowledge the historical graveyard of this specific medical field.
The graveyard of failed drugs.
Exactly. Over the past decade, numerous clinical trials have attempted to combine antiviral agents with immune-stimulating agents, and almost all of them have failed.
I think she noted that only one other phase 3 trial had even been completed before this one.
Right. But she makes a fantastic point about the sheer simplicity of this specific intervention.
How so?
The success of B-Well is remarkable precisely because it achieved these numbers by adding just a single investigational agent, the Bepirovirsen, to the standard daily NA therapy.
It wasn't some incredibly complex, fragile cocktail of 3 or 4 experimental drugs.
Exactly. However, Dr. Locke also anchors the excitement in some pretty harsh public health realities.
Which we have to talk about.
We do. While Bepirovirsen is a highly attractive option for a very specific subset of patients, those without cirrhosis and with low baseline antigens, she emphasizes that curative therapies must ultimately evolve to be accessible.
Because to truly move the needle on a global scale, these therapies need to be simple, they need to be safe, and they have to be affordable.
And let's be real here. A drug that requires intensive biweekly clinical monitoring for severe kidney and liver side effects? That is incredibly difficult to deploy at scale.
Especially globally. If you're trying to reach the 240 million people living with this virus, many of whom are in resource-limited settings, places Without access to high-frequency blood monitoring labs, this current protocol is a logistical nightmare.
It's just not feasible right now for everyone.
Exactly.
Her perspective highlights that we have to view these trials as a stepping stone. It's not the final destination. She hopes these findings will validate the mechanism of action and really encourage the testing of other safer combinations.
The ultimate goal is to drive that functional cure rate much closer to 100% and to stabilize the safety profile so it can be expanded to broader populations, like those with cirrhosis.
Right. Well, let's transition to our analytical science minute, because we keep mentioning the mechanism of action, but we need to explain exactly how this drug pulls the fire alarm.
Yeah, how is this single agent achieving something that decades of previous drugs couldn't even touch?
It requires a fundamental shift in how we think about antiviral medication. Standard NA therapies are small molecules that basically act like wrenches thrown into the gears of the virus's DNA replication machinery.
They just physically stop the virus from copying itself.
Right, but Bepirovirsen doesn't do that. It belongs to a completely different class of drugs known as unconjugated antisense oligonucleotides.
Okay, unconjugated antisense oligonucleotide is a massive mouthful. Let's break that down for the listener, practically speaking. What does antisense actually mean?
To understand antisense, you first have to understand how your cells and the virus build things.
A basic flow of biology.
Exactly. DNA makes RNA and RNA makes proteins. The viral DNA hiding in your liver, that cccDNA we talked about, is the master blueprint.
Okay.
To actually build the viral proteins, including those decoy surface antigens, the DNA sends out messenger RNA. The sequence of that messenger RNA is called the sense, is the readable instructions.
So the messenger RNA is kind of like the foreman on the factory floor holding the active instructions to build the decoys.
That's a great way to put it, yes. An antisense drug is a synthetic strand of nucleic acids designed in a lab to be the exact chemical mirror image of that specific viral RNA sequence.
The mirror image.
Exactly. When you inject Bepirovirsen, it enters the infected liver cell and physically seeks out all the hepatitis B messenger RNA.
And because it's the perfect mirror image, it chemically binds to the viral RNA, like a magnet.
Precisely.
And what happens when they bind? Does it just block the instructions so the factory stop?
It does much more than block them. It destroys them.
Really?
Yes. When this double-stranded pairing occurs, the cell recognizes it as abnormal. It says, wait, RNA isn't supposed to look like this. So the cell deploys an enzyme called RNase H-tol to literally shred the messenger RNA.
Wow.
The blueprint is destroyed before the factory can manufacture a single viral protein. This direct targeted destruction is what causes that rapid, dramatic drop in the viral surface antigens that we see in the clinical data.
It shuts down the decoy factory entirely.
It does.
That is incredible targeting. But earlier, we talked about how it also wakes up the immune system. Shredding the RNA just stops the factory. How does it trigger the immune war?
That is the genius of this specific drug design. It has a secondary, completely distinct effect.
Okay, what is it?
The physical presence of the antisense oligonucleotide itself acts as an alarm bell.
Just its presence in the cell.
Yes. Research suggests it interacts with specific sensors in your immune system called toll-like receptors.
Like toll-like receptors.
Right. These receptors are designed to detect foreign nucleic acids. When Bepirovirsen hits them, it signals to the body's innate immune system that a massive viral infection is present.
It essentially pulls the fire alarm.
Exactly. So the drug is operating on two fronts simultaneously.
First, it strips away the virus's camouflage by shredding the RNA and lowering the surface antigens.
And second, it actively activates the body's immune cells, calling them to the liver to clear out the infected hepatocytes.
It is that dual mechanism, shredding the blueprints while simultaneously calling in the cavalry, that leads to the functional cure.
It's brilliant pharmacology, really.
So, synthesizing all of this, the big picture is really come into sharp focus here.
It is.
For patients managing chronic hepatitis B who do not have severe liver scarring and who have a relatively low baseline antigen level, this 24-week course of Bepirovirsen has proven it can trigger a functional cure in up to 28% of cases.
It represents a highly anticipated, statistically proven exit ramp from the burden of lifelong daily medications.
So if you or someone you know are currently taking daily standard therapy for hepatitis B, There is a very specific, actionable takeaway from this data for you today.
And that is?
The next time you sit down with your hepatologist or your medical provider, explicitly ask them what your specific quantitative HBsAg level is.
That is vital, because a lot of patients only know if they're positive or negative for the virus.
Right. They just get a plus or minus on a chart. They don't know the actual numerical value. But as these new antisense therapies move through the regulatory pipelines over the next few years, knowing your exact quantitative number is going to be huge.
Specifically, knowing if you fall into that critical under 1,000 baseline category.
Exactly. That number might be the defining factor in determining your eligibility for these curative treatments in the near future.
Knowing where you stand on that spectrum gives you a massive advantage in mapping your future care.
And it's important to remember, the medical community is not stopping at 28%.
Not at all. Researchers are already looking at how to utilize the BUL findings to push the boundaries even further.
If Bepirovirsen can achieve this as a single agent added to standard care, the immediate next step has to be combination therapy.
Right. How do we combine this with other weapons in the arsenal?
Exactly. Could combining Bepirovirsen with other targeted immunotherapies or, pairing it with novel gene silencers like small interfering RNAs,
right,
could that push that functional cure rate from 28% closer to 100%?
Or what happens if we sequence it with a short course of that pegylated interferon we mentioned earlier, just to increase the long-term durability of the immune response?
These aren't just hypothetical questions anymore. They are actively being tested right now in upcoming large-scale follow-up studies.
Trials like the Be Sure and Be Together studies.
We are finally moving away from the era of simply handing patients a bucket and telling them to keep bailing water so they don't sink.
The science is now firmly, aggressively focused on how we patch the hole in the boat once and for all.
It's a remarkable shift in the field of virology.
It really is. Well, thank you for joining us for this deep dive as we explored the mechanism and the promise of this new research.
Until next time, keep questioning the data.
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.